So I talked about the structure of PS1, LH1, super complex. Something like this. And then in the last, I just want to show how the pigments, chlorophylls, carotenoids, are organized in the super complex. So this is a view from the stromal side, and these are transmembrane helices. No, these are transmembrane helices, something like this. And this is a PS1 core, okay, reaction center. And we have talked about photosystem too, but this is PS2 core. PS2 core is made of mainly 4 subunits, reaction center. I should say, yeah, core. And PS1 made of mainly 2 large subunits. So the size is different, okay? This is PSAA is about 7 or 80 kilodalton. PSAB is the same size. And PS2 is reaction center is the PSAAD heterodimer, and core antenna PSAC and PSAB are present. That is core antenna. So you may think that the size and also the primary sequence of the amino acid sequence of PSAB and PSABC, PSADB, PS2 and PS1 amino acid sequence very different. It's not very much conserved. But if you look at the arrangement of the transmembrane helices, okay, of PS1 and PS2 are very similar. PS1, PSAA, if you look at PSAA here, it's here. It's N-terminal region, this domain, the 2 domains. N-terminal domain is here. C-terminal domain, C-terminal domain is here. Okay? And also very symmetrically, PSAB is the same. N-terminal domain is here, and C-terminal domain is here. And N-terminal domain contains 6 transmembrane helices. And 2 pair A, B helices, C, D helices, E, F helices have pair. Okay? 3 pair of alpha helix is N-terminal domain. The PSAB N-terminal domain also the same. AB, CD, EF domain, alpha helix. And it's a kind of circular structure. And in the C-terminal domain, 5 helices are present. And 1, 2, 3, 4, 5. And also PSAB, also C-terminal domain is 5 helices. And also, if you look at carefully, the arrangement of this alpha helices is like something like this, here and here, no, like shaking hand. Okay. If you look at the PS2 core, this is reaction center. AD, this is heterodimer, also very similar each other. and 5 helices. And also arrangement of 5 helices like this and like this. So PS1 reaction center component is present here, and PS2 is present here. So these two domain looks similar. And this is a terminal domain of PSA A and PSA B. It's associate antenna, only antenna pigments. So we call this part is core antenna. This part is reaction center. Okay, core antenna, reaction center. And PS2, it's different on the different polypeptide, PSA C and B, core antenna are present. But this arrangement of alpha helix, the pair, 3 pair of alpha helix are circular, like this. So it's similar, this part. Okay, and this contain only core antenna and the reaction center here. So PS1 and PS2, because in the beginning when we determine the amino acid sequence of PS1 subunit and PS2 subunit, they were not similar. So most researchers thought photosystem 1 and photosystem 2 are developed independently from the different origin. But if you look at this 3-dimensional structure, primary structure it's difficult to say PS1 and PS2 are similar. But the 3-dimensional structure are very similar each other. And then specific, some very important amino acid that is involved in associating a pigment or cofactors are conserved. So now we think that the PS1 and the PS2 are evolved from the original, the same ancestral reaction center. But in here, PSA is separated into 2 subunits, and the PSAB was separated. Or I don't know, we don't know, these 2 subunits were fused to a single polypeptide. That happened. And if you look at the number of pigments, PS1, PS2, more chlorophyll molecules you can find in PS1, right? The PS2, no, more spaces. That's different. But PS2 has contain more a lot of light harvesting complex 2, turmeric LHC2 outside the this region is surrounded by turmeric LHC2. So total antenna pigments number is more. You can find more antenna pigments in PS2 unit. Okay. Okay. Another interesting thing is if you look at this structure from the side, the thylakoid membrane, the section, and this is the position of the chlorophyll. This is reaction center. Okay, PS2, this is reaction center. And this is a core antenna. And this is reaction center. And this is core antenna. Okay. So, as I told, if you remember, LHC2, in LHC2, the structure, I told that the pigments, chlorophylls are distributed, appear to be distributed more evenly But if you look at the section of the structure, the chlorophyll is concentrated on stroma side and luminar side, but less chlorophyll in the center. And in the case of the PS1 and the PS2 core, no, this is not a light harvesting. From the stroma side, the pigments are distributed evenly, but if you look at from the side, stroma side, luminar side, pigments are more concentrated, right? In the middle, you don't see many pigments. That is the case in PS2, no? Stroma side, luminar side, and stroma side. More chlorophylls. And in the center, less chlorophyll. Of course, in the reaction center, here, the photochemical reaction occurs. So electron is transferred from the luminar side to stroma side. So the redox component, including chlorophylls, are located across from the luminar side to stroma side. So this is exceptional, right? That is the case. And the photons, for example, PS2 photons, when the light photon absorbed by chlorophylls here or here, and then chlorophyll becomes excited. And this is excited state it's transferred among the chlorophylls, and then finally reach to reaction center. Okay, so probably we think that from the structure, these chlorophylls are closer to the reaction center. So most of excitation energy is first transferred to here, no? And then finally transferred to the reaction center. In the case of PS1, there are some chlorophylls here, but finally, this is more chlorophylls. But the excitation energy transferred from here to reaction center. And one chlorophyll like this is more the closest. Not every chlorophyll, but some specific, two specific chlorophylls are close to the reaction center. So it transfers the excitation energy. That is we can speculate these kind of things from the structure. So this high resolution structure helps us understand how the excitation energy from the antenna to reaction center are transferred. That is the case. So as I told before, even equal distribution, instead of distribution concentration, the luminar astronomer side is make the distance between the chlorophylls shorter. That is very important for efficient excitation energy transfer from chlorophyll to chlorophyll. No, so that is a structure analysis made increased our understanding of the structure function of these pigments. Okay, this is a structure. So conclusion is that these are very organized, precisely organized arrangement of pigments are made possible by protein, no? Scaffold. Proteins arrange and fix these pigments at proper position. So this is precisely done. So there are many subunits are present. So next topic is how this PS1 complex is made. So by particularly the many components, particularly I'm going to focus on these subunits. Many subunits are synthesized independently by translation and then come together, not randomly, how it happens. So this is we call assembly process. Okay, so I want to change the slide to another file. How could someone help me to change? Escape, and then close this, and then LHC lecture. This one. Okay. So this is next is assembly process. Is that okay? Okay. I took from my presentation from some conference, so just slightly different. I show you the highest mountain in Japan, the volcano, Mount Fuji. It's summer, so it's not covered with snow. So, but very our symbolistic mountain. And okay, as we discussed, this is a PS1 LH1 supercomplex of chlamydomonas. And this subunits composition. So in chlamydomonas, PSA, B, C, J are encoded by chloroplast, and the others are encoded by nuclear genome. This is an example of the component composition of vascular plants. This is chlamydomonas and cyanobacteria. This is a prokaryotic. So in the case of chlamydomonas, these are chloroplast and others are nuclear encoded. And the presence of subunits, vascular plants and chlamydomonas, if you compare, there's no PSAM is missing, but in the cyanobacteria it's present. NOP is not found in chlamydomonas, but present in vascular plants, not in cyanobacteria. PSAX is present only in cyanobacteria. PSAGH are present in the vascular plants and chlamydomonas, but it's lacking, deficient in cyanobacteria. So there is some difference in peripheral subunit, but important subunit, PSA A, B, C is associate redox cofactor. So these are all conserved among the cyanobacteria, green algae, vascular plant. And LSC1 is vascular plant has 4 LSC1, as I told you before. And sometimes in some other NAD, PS1 NAD supercomplex, LHC5 or 6 transiently as a minor component is present, but it's usually PS1 contain 4 LHC1. In chlamydomonas, 9 different LHC1 and total 10 copies. The cyanobacteria has no LHC. It contains phycobilisomes. So instead of LHC. So PS1 also accept the excitation energy from phycobilisome that is different from type of the phycobilisome of PS2. So this is different part, peripheral. So but this structure must be assembled properly to express the function of PS1. So this is how PS1 complex is assembled. So if you think about the motor car assembly factory, car is made of, I don't know, 10,000 or many parts, right? But if you just mix these parts and then shake, but you don't find the car, not functional. It's just a mixture of parts, right? So in the factory, what happened is, can you see this? The robot assembling part at the proper position, right? It's not randomly. So and also the order of assembly of parts are fixed, right? If you put the parts in the wrong steps, it's difficult to make cars. Okay, so like like a robot or workers assemble motor car step by step, and then gradually cars are assembled. Okay, and in the final, if you purchase car, of course you don't find in the car the workers, factory workers, or a robot to assemble. No, only you can buy car. It is a very similar step of assembly of complex. There are many components for example, in the PS1 complexes. So probably each subunit assembled into to make a final complex step by step, probably. Okay, from the center to the peripheral parts, probably. No, assembled step by step. We don't know how many steps there are, but and also like factory workers or robot, each parts or each subunit, when they integrate it into the complex, there must be factors that assist assembly. That is called, we call the assembly factors. That is temporarily involved in this process. And when mature PS1 complex was assembled, this factor disappear because we don't find. So to understand how the PS1 complex is assembled, we want to know these assembly steps. And also we want to find assembly factors. Okay. So concerning assembly factors, this is a corpus DNA of chlamydomonas. And we had by chance analyzed two open reading frames. Open reading frame mean there is a initiation codon, the methionine, and then stop codon. But we don't know if this is expressed, transcribed, translated, and we don't know function. So in that case, we named chloroplast hypothetical chloroplast open reading frame. So hypothetical means probably this may be important, may have a function, because in this case, this hypothetical chloroplast open reading frame is conserved in plants, chloroplast, and even cyanobacteria genome. Okay, very conserved. So that must be has function or expressed. So there's a numbering from the large size. For example, it's YCF1, 2, and YCF3 and 4 are present here in chlamydomonas. So hypothetical chloroplast open reading frame 3 and 4. So we by chance knocked out these two genes independently to see a phenotype. Maybe the strategy to do a chloroplast transformation will be I will talk in the other lectures. So we knock out anyway. Then we analyzed the phenotype. Then we found PS1 is gone. Although YCF3 and YCF4 are obviously not a component of PS1, right? But these two are important for accumulation of PS1. So we thought, oh, this may be a factor that assisting or involved in somehow PS1 synthesis. But this was not reported before, so we don't know how this to. Actually, we found that the YCF3 and YCF4 are translated, and proteins present by we detected by immunoblotting. We raised the antibody against the YCF3 and YCF4 and found it's present in the cells, in the chloroplast, in the thylakoid. And if we knock out this, of course, YCF3, but YCF4 is present, PS1 is gone. And YCF3 present, YCF4 knockout, but PS1 is gone. So that's happened. So then we thought that's how YCF3 and YCF4 are involved in PS1 accumulation. Probably we thought at the time already that maybe important for assembly of PS1 complex. But we needed the evidence to show this. So what we did was affinity purification of YCF3. As I discussed before about the chloroplast SRP 43 and ARB 3.1 affinity purification and find out interacting protein. So this is a method. Now, first we transform the chloroplast gene or YCF3 or YCF4 and then HA tagged genes introduced. Okay, and then check that even infusing a HA tag at either N-terminal or C-terminal still works because PS1 is assembled. In the case of chloroplast gene, we don't need to worry about N or C-terminal side. We can insert N-terminal side. So we inserted either N or C and to see which is better. And then we purify this. HA tag is the amino acid is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 9 amino acid. And so we put into the spin column, not small column in the beads that specifically bind the HA tag sequence. So in the mixture of the protein from the cells, HA tagged proteins and the cross is cellular proteins, the mixture, and then we add the beads, the antibody against HA tag, mixed it, and then this part is put into a little bit small or larger tube, and then spin down. Okay, so cross is a cellular protein without HA tag is eluted out, and beads associating HA tag protein remain here. Is that clear? You can see. So if you do this washing few times, then you can completely remove the protein without HA tag. And then peptide solution containing this peptide, synthetic peptide. Then HA tagged protein is eluted out. And then these beads associate this synthetic peptide because concentration of synthetic peptide is much, much or higher, right? So you can replace this. Then you can purify this HA tagged protein. Okay. So YCF3 HA affinity purification. This is the result. We use wild type as a negative control. You don't see practically nothing. But there may be some band here. But YCF3HA, you can purify here. And a little bit smaller contamination, similar contamination. And also, there's another band here. Okay, so pure, I should say, because the amount of YCF3 is catalytic amount. Okay, so even though we can purify very nice band here, and then may be minor, but if you look at the intensity, compare the intensity, it's almost no contamination, practically no contamination. Concerning this interacting protein, some German group, Arbas, also purified using a tobacco. Their result is here. This is negative control. They use a flag tag. So even negative control, there are many proteins like here. And this is positive control. Positive, this is the YCF3 flag. So YCF3 was here. And others are, these are all non-specific contamination. And then they also find a specific protein that is lacking in the negative control, but here. So they named it YCF3 interacting protein 1, Y3IP1. But we found this, also did this experiment with later, published 8 years later, but our preparation is much cleaner. This was done in the Nepalese leader from this Hyderabad. He was a postdoc in my laboratory, and then he did this experiment. Huh? Sridhar. So what is nice compared to this, purity is high. It's just looks nice. Not only looks nice, but it's very useful. If you detect it, the major protein is these two, almost stoichiometrical amount. But if you detect it, the immunoblotting, this is Y type. You don't see any. But this YCF3 tagged purification, of course, YCF3 here, tagged, and Y3IP1, and a little bit YCF4. You don't see here the band, but it's what's present. And what is important is small amount of PSA A, F, but not other. PS1 subunit was small amount of PS1 subunit was there. But if you use this kind of not so pure preparation, if you see polypeptide other than YCF3, nobody believes that is authentic interacting protein, but it's maybe contamination. Okay, so since the YCF3 is essential for PS1 accumulation, we expect it's possible that the YCF3 has somehow had interaction with PS1 subunit to assist PS1 accumulation. That is, to assist the PS1 assembly. So to assist assembly, the factor must somehow interact with PS1. As the motor car, as a car assembly factory, robot catch one parts and put into the car. So there's interaction, right? The same thing happened. So this immunoblotting data suggests this interaction. But of course, there's a possibility that this interaction with PS1 is non-specific contamination, non-specific interaction. Okay. So what we did was radio labeling experiment. I told you before that newly synthesized proteins, after pulse labeling, newly synthesized proteins are labeled. Okay, after chase, pulse and chase experiment, then if the interaction is stable, a labeled protein stays there. But interaction is transient, radio-labeled protein disappear. Okay, so we checked. We labeled protein with S35. So sulfur is better than carbon-14 to detect, more sensitive. So this is a thyroid membrane. Pulse labeling, C is chase. Pulse labeling, short minutes, 1 or 5 or 10 minutes. Chase is 1 hour or 2 hours, something like that. Okay. In the thyroid membrane, you see a labeled protein like this. And after pulse and chase, you don't see much difference in a labeling. See, that means on the thyroid membrane, the pulse labeled protein is present, and also even after 2 hours, this protein stay in the thyroid membrane, as expected, because synthesized protein is present in thyroid membrane, not after its synthesis. That is as expected, nothing special. So it's actually this ATP synthase alpha and beta subunit, and this I don't know exactly, but this is probably LSC proteins. Okay, because this time pulse labeling was not 1 minute; it's a little bit longer. So LSC is proteins labeled. Okay, from this pulse labeled and the chase labeled, pulse labeled chase thylakoid membrane, we purified YCF3 the same way, but using labeled thylakoid. Then what happened? Okay, so YCF3 HA tag here, the PSAF here, PSAF was a little bit present. You can detect, okay? And also there's many diffuse band, but YCF3, YCIP1 was somewhere like here, okay? According to the position. And some proteins are there. But what is important is PS1 reaction center proteins, PSA A and B, labeled proteins were present. Okay, reaction center protein. We couldn't detect by staining because there are some diffuse band around here, and also amount is so low. But label was clearly shown. And after chase, reaction center protein, this diffuse band, disappeared. And also PSAF was also disappeared. Somehow, YCF3 also labeled YCF3 disappeared. Y3IP1 present. The others are also very present. So this means that the PSA A and B newly synthesized PSA A and B are transiently bound to YCF3. And also YCF3 itself turnover. This means turnover. This is newly synthesized and it's disappeared, digested. But Y3IP1 is stable. Not much labeled, but it stays. The stoichiometrical amount, but this turnover is fast and this is slow. But still, they present as dimer or oligomer, but stoichiometrical amount according to the staining band. So, so this data clearly show that YCF3 is associated with newly synthesized PS1 protein. So which this result is consistent with YCF3 its assembly factor, because assembly factor interact with newly synthesized PS1 protein. Okay? So this data is clearly show that YCF3 is one of the assembly factor of PS1. Okay. But this is more related to only reaction center protein, or PSAF a little bit, but mostly, but other PS1 subunit were not interacting. So from this data, we conclude that mainly PSA A and B, assembly of PSA A and B reaction center of PS1 is more core part. It's an important part. In this assembly, YCS3 is involved. So this is a very initial steps of PS1 complex assembly. And also Y3IP1, YCF3 interacting protein, what this does. So Y3IP1 minus mutant, also in the chlamydomas CGL51. CGL1 means chloroplast green lineage, so it's related to some photosynthesis or something. Number 59. This is another name of Y3IP1. So we obtain the clip library of Chlamydomonas center. So we can obtain by paying $100 US dollar. So we can get this this delta Y3IP1 mutant. Okay, the YCF3 mutant, YCF4 mutant. We check the growth of the cell. This is a dark condition, but on the top media. Top media contain taurus acetate phosphate. It contains acetate. That is a carbon source for respiration. So chlamycell can grow heterotrophically under this condition in the dark. So these 4 strains grow well. Okay. And if you illuminate here 50 micromole photons per square meter per second, wild type grow better. But YCF3, YCF4 very photosensitive, so it didn't grow. Y3IP1 a little bit grow, very much reduced, but it's still better than YCF3 and YCF4. And this is a HSM media. Minimal high salt minimum. This lacks acetate. Okay. So cell can grow only under the photoautotrophic condition. So Y3IP1, YCF3, YCF4 didn't grow at all. Okay. So Y3IP1 is also very important for photosynthesis, probably PS1. And we also checked these 3 strains by immunoblotting the presence of related proteins. This is wild type, controlled wild type. We loaded 100%, 30, 10% of the sample. This is a dilution series. And also PSA-A, YCP1, YCP3, YCP4, you don't see the signal, but very, very slightly delta Y3IP1 mutant accumulate PS1. Okay. You can see it's less than 10%, maybe 5%, something like that. And also Y3IP1 present, but this mutant lacks, of course. YCF3, YC3 mutant lacks, but Y3IP1 still accumulate YC3. YCF4 mutant accumulate YC4. YC4 mutant lacks YC4, but others are still present. So, because we detected very, very small amount of PS1, and also photosensitivity is a little bit less than delta YC3 and YC4. So we expected that there may be small amount of PS1 is assembled. So we isolated thyroid membrane, solubilized with mild detergent, and separated extract by circumstantial gradient, as shown many times. This is the top of the gradient and the bottom of gradient. It's for Y type. We can see LHC2, PS2, PS1 LHC super complex. Nice 3 green band. And from YCF3 mutant lacks, that which lacks PS1, it's only LHC2 and the PS2 and the LHC1. Without PS1, LHC1 accumulates, so 2 green band. From the delta Y3IP1, that accumulate a small amount of PS1, 2 band, but slightly green band are present in PS1 LH1 super complex. The presence of small amount of YPSA1 in this mutant mean that it's assembled. not the protein present or protein, but it's assembled. So we took this fraction also from wild type and from this mutant. Also we checked blotting of this fraction and checked PSA, C, D, L, LSC4, LSC. So the PS1 and LSC1 protein was detected by blotting. Although the amount is lower, but still present, it seems that its assembly is almost perfectly worked. Then we isolated this fraction. We measured PS1 activity. Okay, so the wild type show when you illuminate P700 or PS1 reaction center oxidized, and then you stop the illumination, it's reduced. So it automatically. And delta Y3IP1, this is small amount of PS1, so it's also show activity. So it's assembled. So I thought I mentioned that the Y3IP1 is important for PS1 accumulation, but this shows that it's not essential. Without Y3IP1, PS1 is assembled in a very inefficient manner, but still possible. So we said, because Y3 is present already here. So what happened? So essential component is Y3.
Sorry to disturb you. It's not related to the topic we are talking, but I have a doubt in the Western blot. Yeah, Western blot in the second figure. Yeah, actually for mutant confirmation, no, the top right side, second figure, Western blot.
This one?
Immunoblot, yeah. That figure for mutants, exactly we cannot see the respective bands, right? YCF3.
YCF3.
Yeah, mutants we are not able to see. Respective mutants we are not getting the bands. No. I mean to say the respective mutants. Let's take Y3IP1 is a mutant, right? Yeah, in that the expected protein signal was not there, right? So this is antibody generated specifically by you, or it's like commercially available antibody
you mean this one?
Yeah, down. I mean the Y3IP1 is there, right? The second line, yeah. In the mutant there was no band. That's right. That's correct.
This is mutant.
Yeah, but the antibody is specifically generated, or it's like commercially available antibody?
This is specifically generated from the by overexpression of E. coli.
Okay, yeah. Otherwise, again, not specific. They may bind somewhere else also, no?
This is a specific.
Because I do have the mutant. So when I thought of doing Western blot, this question came. So if somewhere else they would have designed the antigen, they may still bind, even though if the insertion is there. Still they could signal.
You mean the small truncated proteins?
Still they give a signal. Yeah. But I think you should
but anyway, we couldn't detect anywhere. Of course, there's the possibility small amount, but it's maybe there's small possibility, but
because I want to do that. So I had a doubt whether it should be specific. Then I should design the antibody like you specifically. Then only I can get the expected results.
This was probably, I don't remember, it's a synthetic peptide or overexpression. Probably this is synthetic peptide.
Okay, yeah.
So of course
it should be specific to the site of insertion. Then only the cleavage will disturb the sequence, so we may not be able to get the
in the middle of the coding sequence.
Yeah,
the step marker is inserted. So usually protein will not be expressed. This possibility is I don't know. We thought it's unlikely. Of course, in the 100%, but 99% unlikely. Okay, that's it's always good to suspect data or possibility. So now the question is, this is knockout mutant, but in the part of the Y3IP1 may be expressed and translated and present in somewhere in a small amount, but that couldn't detect by immunoblotting, and that also had assembly of PS1. But yeah, when we submitted paper, no reviewer complained about this. So we just possibility. Yeah, okay. Always this kind of question is important to ask. So Y3IP1 and YCF3, this is additional YCF4. But we could detect YCF4, but this comes and then assembly occurs. And this YCF3 turnover the same rate as PS1, so that means we thought it's unlikely, but the YCF3 is once it's assembled PS1 one time, it disappears, and new YCF3 is synthesized. That is waste of energy, but it's according to the pulse chase labeling experiment, that may happen. So, but we don't know why this is important, this turnover. This is unexpected data. This is until 2. Oh, finishing? Oh, finishing.
We can wait for.
Okay, so I will stop. I will talk this to be continued tomorrow. Okay. Any question? Other question? Sorry for. There are so many detailed data, so it takes time to explain very well. So, is there any? Yeah. Could you use a microphone?
Okay, my question is from the first part. PS1 core has more pigment molecules, you showed, right? PS1 core, more pigment molecules, more chlorophyll molecules arranged.
Yeah, a structure.
So when photoinhibition happens in highlight stress or something, it affects more PS2.
Turnover is PS2, yeah.
PS2. So if more excited chlorophyll molecules, there must be chance for more ROS production in PS1. So is that this packed arrangement of pigment molecules is helping its efficient energy transfer, and so that the ROS production is not happening, or why it is like more protected than PS2?
I think PS2. Okay, it's a question. It's a PS. There are the two photosystem, PS1 and PS2, and under the highlight condition, PS2 is preferentially photo damaged by, and then that is photosystem 2 has the system to repair the damaged PS2. Then once the stressful light is removed, it's very quickly PS2 is recovered, right? The PS1 has no such a system. So once PS1 is damaged, there's no repairing system. So the PS1 must be synthesized. Whole complex must be synthesized. So it takes time, and the photo damage of PS1 is affected very more significantly. So basically PS2 photo damage is important for protecting PS1, right? PS2 supply electrons to PS1. So, but under strong light, if PS1 works, it generates a lot of reactive oxygen species. So if PS2 is the damaged, the electron will not be provided to PS1. So without electron, PS1 cannot maybe generate much less reactive oxygen species. Okay. So to avoid photo damage of PS1, donor side of PS1 must be oxidized. So that means that the reducing side of PS1 is reduced, over-reduced. That generate reactive oxygen species. So to avoid this, there's a cyclic electron transfer around PS1 to avoid the over-reduction of reducing side of PS1. And also PS2 is blocked, then electron will not come to PS1, so PS1 cannot work without the electron. And also, if the luminal side is over-reduced, acidified, the luminal side acidified, so then photosynthetic control occurs. That is, I haven't mentioned this. The strong acidic condition, electron transfer from cytochrome B6F to PS1 is suppressed. But there are many safety valves for not producing reactive oxygen by PS1, I think.
And also, you think this pigment molecules arrangement is making it more efficient for the excitation energy transfer in PS1?
In PS1, yeah, more than PS2. I don't know. That is both are very efficient, but PS2 quantum efficiency is lower than PS1. So PS1 is very efficient. Quantum efficiency is almost 1.
Yes.
So the PS1 in synthesis is how to say, it's not involved in photo inhibition compared to PS2. So this is a very important point. Is that right? Right, correct answer? My answer is
yes.
Okay.
Thank you.
Any question? No, don't hesitate to ask me anything. That if you have a misunderstanding, that is very good to understand better, and also maybe others also the same question. So it's good to have give questions and the answer that helps understanding a lot. But maybe later. So maybe we are hungry. Okay, thank you. So we continue tomorrow.